Welcome to the deep dive. Today, we're taking a deep breath and a deep dive into the future of genetic medicine. For years, we've used these broad genetic risk scores to estimate, your susceptibility to diseases. But that approach, it has always felt a little blunt.
Oh, it is blank. We're moving beyond the wide brushstroke and picking up a scalpel.
Okay.
We are examining a groundbreaking new framework called scPRS, that's single-cell polygenic risk scores, which promises to tie genetic risk not just to you, the individual, but to the specific critical cells driving your disease.
That sounds incredibly powerful. So let's start with the baseline for everyone listening. When we talk about standard genetic prediction, we're talking about the standard polygenic risk score, or PRS. What's the fundamental idea there?
Well, PRS is really designed to predict your genetic risk for complex common diseases. So things like type 2 diabetes or T2D or maybe Alzheimer's disease. It's an aggregation of risk.
An aggregation? And how is that calculated, in the traditional sense?
Researchers survey your entire genome. They look for thousands of common genetic variants, the ones present in, say, more than 5% of the population.
Okay.
Then they calculate your risk by just adding up the weight of each of those variants. And that weight is based on how strongly that variant was linked to the disease in these massive population studies, the Genome-Wide Association Studies, or GWAS.
So it's a linear model. You're just summing up thousands of tiny effects across my entire genetic blueprint.
Exactly.
That seems comprehensive, but we know it often falls short of explaining the actual biology. If PRS tells me I have a high genetic risk for heart disease, why can't it tell me why?
Because of something called cellular heterogeneity. A complex disease. I mean, it doesn't hit every cell in your body equally, right?
Great.
Your pancreas has alpha cells and beta cells. Your heart has cardiomyocytes, structural cells. And the genetic risk variants, especially the non-coding ones that don't change proteins but just control when and where genes get turned on.
Just switches.
The switches. They influence disease in these highly specific, cell-type specific ways. Traditional PRS just averages that signal across all your cell types, and it blurs the biological picture. It's an accurate probability, maybe, but it's a terrible mechanism map.
So it tells you your house is at risk of flooding, but it doesn't tell you if the leak is in the basement or the attic.
That's a perfect analogy. Exactly. Yeah. And that lack of resolution, it limits both its predictive accuracy and, frankly, its utility for developing targeted drugs.
Which brings us to scPRS. This uses a graph neural network, a GNN, and it attempts to compute risk at the level of the individual cell. So what is the core data input that lets scPRS get to this new level of resolution?
We needed a way to see what the DNA was doing in each specific cell. So we use single-cell ATAC-seq or scATAC-seq.
Okay, what is that?
This technology, it maps chromatin accessibility. Think of your DNA as this giant, tightly wound archive. This tech tells us which regulatory regions, we call them candidate cis regulatory elements, or CCREs, are currently open and available to be read in that exact cell.
So if a genetic risk variant sits in a piece of DNA that is only open in, say, a neuron, scPRS can filter out all the noise from the other 100 cell types in the body where that variant is totally irrelevant.
Precisely. We focus on genetic variants only if they fall within those specific open CCREs. This is the first big step. It's called deconvolution.
Okay.
We calculate a kind of microscore for that specific cell type, only using the variants that are actually active there. And this is why scPRS can use far less data, often less than 11% of the total variance from a standard GAVA, yet get superior predictive power.
Wow.
It's all about context, quality over sheer volume.
That makes the signal localized, I get that, but
Yeah.
I know single-cell data can be messy. It's often sparse. You get data loss, right? How does the GNN, the machine learning part, how does that fix the problem?
Right, that's the second step, GNN refinement. Because the data is noisy, we leverage the power of the graph neural network. The GNN effectively builds a map showing how similar all the measured cells are to each other based on their genetic and epigenetic profiles.
So like connecting all the workers in a factory who perform the exact same job.
Yes, that's a good way to put it. If we have a sparsely measured beta cell, the GNN uses that similarity map to borrow information, to smooth and denoise the signal from other very similar beta cells. This captures these complex, nonlinear relationships that a simple linear model would just completely miss. It makes the signal much clearer before we aggregate all those individual cell scores to get a final, more accurate disease risk prediction.
Okay, wait, let me challenge that for a moment. If the GNN is essentially pooling wisdom from like-minded cells, is there a risk of it smoothing out a truly unique or rare signal? What if the most important cell is the one that looks slightly different from all the rest?
That is a crucial question. And the benefit of the GNN is that it assigns an importance score, what we call learned weights, to each cell type in the network. So while it uses neighbors for that initial denoising, the final model prioritizes those cells that consistently drive the highest predictive value.
Ah, I see.
If a rare cell type has an extremely high importance score, the model flags it. This internal interpretability is key because those importance scores tell us exactly which cell types are the most genetically relevant contributors to the disease.
Okay, that clarifies the whole mechanism. So, to recap for you at home, standard PRS is limited by cellular complexity.
Right, it oversimplifies.
Then scPRS comes in, filters that data using the cell-specific open DNA regions, the CCREs, and then uses a smart network, the GNN, to clean up and weigh that signal.
And the result is a highly specific risk score, and crucially, an interpretable biological map.
So, if the vast majority of our genetic risk comes from these non-coding regions acting in a highly cell-specific way, does that change where we should focus our preventative efforts?
And it has to.
Okay, moving on to performance. Did this complex new architecture actually lead to better predictions than the existing algorithms?
Oh, absolutely. The framework was benchmarked against several standard and quite sophisticated prediction methods. It showed superior performance, and we measure that by AROC and AUPRC in 4 test diseases: hypertrophic cardiomyopathy, HTM, Alzheimer disease, AD, and severe COVID-19.
You said 4 diseases, but you only listed 3. What was the nuance in the 4th one? Type 2 diabetes.
Ah, right. T2D was the exception where the initial scPRS model was only comparable to the baselines, not better.
And why was that?
The reason is that T2D risk isn't exclusively driven by changes in gene regulation that you can see through chromatin accessibility. It's also strongly influenced by other variant types.
Like what?
Like coding variants that actually change the protein itself, or splicing variants that alter how the gene is assembled. These aren't visible in the open chromatin data alone.
So the model was blind to variants that might affect the finished protein structure, rather than just the switch that turns the gene on and off.
Precisely. So when the researchers integrated a score capturing those non-accessibility variants, they created the scPRS Plus model. The performance for T2D boosted significantly. It actually outperformed all the baselines then.
Well, it taught them that for a disease like T2D, you really need an integrated model.
You do.
Now, here's where it gets really interesting for me. The interpretability. Tell us what scPRS discovered about the actual cellular drivers of these diseases.
This is the exciting part. By analyzing those important scores, the learned weights, the model pinpointed the critical cell types.
Okay, give us an example.
Look at T2D in the pancreas. Traditional research focused almost entirely on the insulin-producing beta cells. scPRS confirmed the role of the ING beta cells, but it also strongly prioritized GCG alpha cells, which produce glucagon. This confirms that T2D is really a dual-cell regulation problem. The failure to manage glucose involves both the insulin and the glucagon-producing cells. It's a two-sided coin.
That refines the target hugely. What about the heart condition, hypertrophic cardiomyopathy, HCM? What was the novel discovery there?
HCM was striking. The model, of course, confirmed cardiomyocytes, the main heart muscle cells, as key players. No surprise there.
Right.
But it also significantly enriched parasites in the left ventricle. Parasites are structural cells that regulate blood flow and vascular homeostasis. And crucially, established, less precise methods had failed to identify parasites as major genetic drivers.
So scPRS flagged a completely new biological candidate for HCM drug targets.
It did, thanks to its high resolution.
So we potentially found a new source of disease vulnerability in the supporting structure of the heart muscle.
Okay, what about Alzheimer's disease?
The AD findings, they really validated and expanded our understanding of the brain pathology. scPRS strongly prioritized microglia, the brain's immune cleanup crew, as well as astrocytes and oligodendrocyte progenitor cells.
OPCs.
Right. And this just reinforces that AD is fundamentally a disease of complex glial dysfunction. It's not just about dying neurons.
And for severe COVID-19, I think I can guess this one. You probably can. Unsurprisingly, it prioritized the immune responders in the lung: macrophages, natural killer cells, and monocytes. It validates their role in driving that inflammatory cytokine storm that leads to the most severe outcomes.
It really is the difference between knowing the risk and knowing the reason. So if we can identify which specific cell types are genetically driving a disease with this level of granularity, how should that knowledge change the way we approach drug development? I mean, should we be aiming for cell type specific delivery?
I think we have to be. And that leads perfectly into the final application. mechanistic dissection. Once scPRS flags a cell type, the next step is to find the specific variant, the specific piece of open DNA, and the specific gene it controls.
The trio.
The trio. They use a sophisticated computational pipeline, a layered multi-omic strategy, to map these causal variant CCRE gene trios.
Okay, let's turn this abstract pipeline into a detective story. Start with T2D and this aging beta cell connection. Which genetic variant were they tracking?
They focused on a T2D-associated variant called RS108111660. scPRS pointed them to an A and she beta cell-specific CCRE.
So a regulatory region only open in those specific beta cells.
Correct. And computational predictions suggested the risk allele actually reduced the accessibility of that regulatory switch.
So if the switch is less accessible, what gene is affected?
Well, they used bioinformatics tools to find co-regulated genes and link that CCRE to a gene called CDKN2AA. That gene is critical because it encodes a protein, P16INK4A, which is known to restrict beta cell proliferation and insulin secretion as we age.
That sounds like a really bad thing for a cell that's already overworked in a diabetic patient.
Exactly. The proposed mechanism is that the T2D risk allele causes the binding of a key transcription factor, alpha, which in turn upregulates CDKN2A sesmeration in those beta cells.
Wow.
Higher CDKN2A expression means accelerated aging and restricted function in the insulin-producing cells, directly increasing T2D risk. It's this clean, beautiful link between genetic risk, cellular aging, and disease, all isolated to a specific beta cell subtype.
Let's move to Alzheimer's. You mentioned scPRS can prioritize variants that traditional GLAers often miss because they fall below that strict significance threshold. Give us an example.
The variant RS7922621 was one such nominally significant variant. Traditional studies might just ignore these, but scPRS focused attention on it.
Why?
Because it only affected chromatin accessibility and microglia, those brain cleanup cells.
So what was the functional consequence inside the microglia?
The variant was predicted to regulate two specific genes, ANC11101 and SBN1490. And to prove this wasn't just a prediction, the researchers went into the lab. They experimentally suppressed those two genes in microglia-like cells that were derived from stem cells.
And what happened when they silenced those genes?
When ANX11 and TSPAN14 were suppressed, the cells showed a significant impairment in microglial phagocytosis.
Their ability to engulf and clear away junk?
Right. Their ability to clear cellular debris and those harmful amyloid plaques. So, the risk allele for AD reduces the gene expression, which suppresses the cleanup function, thereby raising AD risk. And this is the ultimate validation. scPRS pointed to the suspect cell, it identified the target genes, and then lab work proved the functional defect.
That's incredible.
So scPRS is truly a unified framework. It doesn't just give you a better prediction score, it gives you an actionable blueprint. The key is its ability to weigh the genetic importance of each cell type, overcoming the resolution limits of those older bulk-level methods.
And remember that number.
Right. Superior predictive power, often achieved using less than 11% of the total available variants. It's a testament to the power of context.
That efficiency is just staggering. It really highlights that the context, knowing which cell and which regulatory region, is far more important than just amassing raw data. So what are the next steps for this technology? What's missing?
Well, the current model relies mainly on chromatin accessibility data. And as we saw with T2D, where coding or splicing variants are critical, focusing on open DNA alone isn't always sufficient.
Right.
Future developments have to integrate other single-cell modalities like data on gene expression from cRNA-seq or maybe DNA methylation. This integration will provide a complete multi-layered view of how genetic risk is expressed molecularly across all cell types, and that will further enhance both prediction and discovery power.
This research really reshapes the map of personalized risk. It moves us from these general genetic probabilities to pinpointing cellular defects. It's an incredibly exciting step toward true personalized medicine.
It allows us to ask far more sophisticated questions about how diseases start and how they progress.
And that leads us to our final thought for you, the listener. If we can identify, at the level of a single cell, the exact genetic pathway driving your personal risk for a disease like HCM, the specific parasite pathways, for example, how should that highly individualized cell-level knowledge fundamentally change the way your condition is monitored and treated? Will future therapies be micro-targeted directly to the single specific cell subtype responsible for the problem?